Hello, everyone. My name is Bryan Reatini, and today I'm going to be talking about some of the work that I've been doing at the University of Arizona for my postdoc. Specifically, I'll be talking about generating genomic resources for yellow starthistle and using them to investigate the genetic basis of traits contributing to its invasion. Yellow starthistle is a diploid species belonging to the family Asteraceae that has a broad native range throughout the Mediterranean and is invasive in the western United States as well as Chile and Argentina. Yellow starthistle was originally introduced from Western Europe to South America in the 1600s, followed by secondary introduction from Chile to the Western US as early as 1860. Starthistle is a common weed in alfalfa fields in its native range, and it appears that it was introduced to the Americas by hitchhiking along with shipments of alfalfa. And given this well-documented history of its introduction and the considerable ecological and economic impact that this invasive species has in the invaded range, Yellow Starthistle has become a model system for studying the causes and consequences of species invasions. And one of the really interesting observations that has come from studies on invasive yellow starthistle populations is that when invader genotypes are grown alongside native genotypes in a common garden, invaders tend to grow larger on average. And this is important because it appears that larger size allows for greater investment in reproduction once these individuals bolt, which in turn is predicted to lead to greater population growth rates for invader genotypes relative to native genotypes. So this trait, which appears to be underpinned by differences between these genotypes, may be contributing to the rate of spread of this invasive species. And this makes this trait a great candidate for investigating how evolution can contribute to rapid spread of invasive species. The problem, though, is that as with many invasive species, which tend to be non-model organisms, genomic resources for yellow starthistle are currently limited. And so the first step in investigating the genetic basis of increased size in the species was to generate the necessary genomic resources, namely a reference genome, annotation, and genetic map. For the reference assembly itself, we shipped DNA off to Dovetail Genomics. Assembly involved the construction of a draft assembly using PacBio hi-fi reads, and then subsequent scaffolding using proximity ligation data via Dovetail's Omni-C technology, which is a spin on standard Hi-C. And the resulting assembly had a total length of roughly 765 megabase pairs, which is roughly 84% of the average genome size for this species, estimated by flow cytometry. And 93% of the total length of the final assembly was in these first 8 scaffolds, with all of the trailing scaffolds being less than a megabase in length, as we can see here in the size distribution plot, where the final assembly is shown in red. And the importance of the L90 of the assembly being 8 is that the haploid number for this species is 8. So this suggests that these 8 primary scaffolds, which cover over 90% of the total sequence content of the assembly, likely represent the 8 haploid chromosomes of the species. In addition, 93% of the benchmarking single copy orthologs in the embryophyta busco dataset were found to be complete within just those first 8 primary scaffolds, and no additional buscos were recovered in the trailing scaffolds, although some were duplicated within them. And this again suggests that the 8 primary scaffolds alone offer a fairly complete representation of the genome of yellow starthistle. In order to further validate the assembly, I then constructed a genetic map from an F2 mapping population produced between a native and invader genotype using RAD-seq data. And specifically, in order to validate the assembly, I asked first whether the linkage groups within the genetic map corresponded with the putative chromosomes of the assembly, those first 8 scaffolds. Second, whether the genetic distances between the loci in the genetic map were consistent with their physical positions within the assembly. And what I found is that each of the 8 linkage groups in the genetic map did indeed correspond with one of the 8 putative chromosomes in the assembly, as we can see here where I've mapped the physical position of the markers from the genetic map onto the first 8 chromosomes of the assembly, with each linkage group displayed as a different color here. So yes, the linkage groups in the genetic map do correspond with the putative chromosomes in our assembly, but what about the collinearity between the genetic map and the assembly? When we compare the genetic and physical maps of chromosomes, there are a few patterns that we might expect to see. First, if physical linkage between markers is the only thing contributing to, the only thing influencing recombination rates, we would expect to see a 1-to-1 relationship between the genetic map and physical, or between genetic and physical distance. However, in reality, we know that the landscape of recombination is not typically uniform across the chromosome. A number of features can reduce recombination, such as proximity of markers to the centromere, which can skew the relationship between the genetic distance and physical distance between markers, such that genetic distance in the genetic map doesn't progress in these regions of low recombination, whereas physical distance does. And when we look for these patterns in our data, we do indeed find them. What I'm showing here is the collinearity between the genetic map on the y-axis here and the physical distance of each of the chromosomes on the x-axis for each of those 8 putative chromosomes of the assembly, with putative centromeric regions marked by these gray vertical bars. I estimated the location of those centromeres using genome scans of gene density shown here in blue for each of these chromosomes and repeat density in black, with the expectation here being that the density of these elements should be lower in pericentromeric regions. And across these 8 chromosomes, we see a few patterns that I'd like to draw our eyes to. First, for 4 of the 8 chromosomes, we see that expected pattern that we outlined before, where recombination is high on towards the ends of the arms of the chromosome, and recombination is low in pericentromeric regions. For 2 of the other chromosomes, we see recombination on only one of the 2 arms of the chromosome, and we do not see recombination on the other arm. And for these 2 chromosomes, it appears that this is because the arms themselves are highly asymmetric. You'll notice that the putative centromeric regions for these 2 are skewed towards one end of the chromosome, such that we do not see recombination on the short arms of these 2 chromosomes. For the last 2 chromosomes, we similarly see low recombination on one of the 2 arms, but in the case of these 2 chromosomes, it doesn't appear to be explained by the position of the centromeres. Both of these chromosomes have fairly centrally located centromeres, and so it doesn't appear to be centromeres that are causing this low recombination across these larger arms. So what might be reducing recombination in such a large region on these 2 chromosomes? I'm not certain yet, but one thing that could potentially explain this pattern is structural variation. For example, there could be large-scale inversions on the arms of these chromosomes, which would similarly reduce recombination in the genetic map. So overall, we do see the patterns that we expect to see from our collinearity analysis between the genetic map and genome assembly, and this agreement between the 2 suggests that the linear sequence of each chromosome is fairly well assembled. And for the sake of brevity, I'm not going to discuss the analysis and validation of our annotation for this genome here, but I'd be happy to show the results of my gene sentinel analyses after this presentation, if anybody is interested or has any questions. Nevertheless, with the genetic map and assembly in hand, we can finally start to investigate regions of the genome that are associated with various invasion traits. And just as a reminder, the trait of interest for this particular species here is size. The invader genotypes are typically larger on average, and as a result, they have greater reproductive output. And the way that this has been measured in previous studies includes a few specific phenotypic measurements, including the total number of leaves, and then the maximum leaf width and length for that particular plant. So for the F2 mapping population, 2 of these components of plant size, namely the number of leaves and maximum leaf length, were measured for all individuals at 2 different time points, because these measurements can differ between seedlings and pre-reproductive adults, between the populations, that is. But what we see for the parental generation here in the mapping population is what we would expect given previous work. Namely, the invader genotypes are larger than the native genotypes for both traits, total number of leaves, and maximum leaf length at both time points. Interestingly, the F1 individuals tended to be larger than either parental genotype, displaying heterosis for max leaf length at both time points and for total number of leaves at the second time point. And then finally, F2 individuals display the greatest range in these trait measurements. So there was quite a lot of phenotypic variation to work with in downstream QTL analysis. I carried out QTL genome scans using RQTL2, using both haley-knot regression and the leave one chromosome out approach. The benefit of the latter, the LOCO approach, being that it attempts to account for kinship for crosses between divergent populations, which was the case for our mapping population. Nevertheless, I carried out both of these methods because at this stage in this work, we're searching for candidate regions to investigate further. So we wanted to be fairly conservative with how we were calling potential QTL. And for the same reason, I used 2 different significance thresholds to call QTL peaks, the standard significance threshold of 0.05 and a suggestive threshold of 0.1. And using this method, I identified a total of 13 QTL peaks, 7 associated with maximum leaf length alone, 4 associated with total number of leaves alone, and 2 associated with both of these traits. For all 4 peaks associated with total number of leaves, the dominance pattern was additive, and the invader alleles at each QTL were associated with greater number of leaves. For the 7 peaks associated with maximum leaf length alone, we observed mixed patterns of dominance, and the parental allele driving larger leaves actually varied between the loci. And by comparing these peaks with the collinearity analyses we looked at before, we can pick up on a couple of interesting patterns. Just to orient you to this plot here, panels A and B are the collinearity analyses and centromere genome scans that we were just looking at a little, shortly prior. Panel C is the loco genome scan for maximum leaf length, and panel D is the same for total number of leaves. The black and green curves in these plots are the results of the scans themselves at time point 1 and 2 respectively, and the horizontal solid and dotted lines are the 0.5 and 0.1 significance thresholds for these scans. One of the things I'd like to draw your eyes to here is that by using these plots, we can see that some of these peaks are going to be more informative than others. For example, the 2 peaks on chromosome 2 here are fairly narrow, and so there is a smaller list of candidate genes within these peaks to sift through to evaluate functional variation that might be contributing to size differences, whereas this peak on chromosome 4 corresponds with that region of low recombination in the pericentromeric region, and this peak is probably going to be a little bit less informative given that the genes contributing to this trait could be anywhere within that 50 megabase pair region. But the peak that I would really like to focus on here is this one on chromosome 1. This peak corresponds with that same region of low recombination that we identified in the collinearity analysis, that kind of odd region that didn't appear to be explained by the centromere location, and the one that could be something like structural variation, such as an inversion. And this putative region that could be an inversion, which corresponds with this QTL peak, spans 88 megabase pairs of that chromosome, and it displays both pleiotropy and overdominance, with heterozygotes being the largest of the 3 genotypes for that peak. Now remember that F1s display heterosis for both traits, which to me raised the question of whether there might just simply be many heterotic genes contributing to both of these traits that are acting as a single locus because they're locked within this inversion, if it is indeed an inversion. If so, that raises an even more interesting population genetic question, which is, might balancing selection be acting on heterotic structural variation, and could that be contributing to increased size within invasive yellow star thistle populations in nature? This, of course, is speculative at this point, but it offers a potential interesting thread to investigate in future work. And with that, I'd like to thank my collaborators who've been working on this work with me, lab members of the Barker and Dlugosch lab, which have helped give feedback throughout this process, and of course, my funding sources, NSF, for making this work possible. And with that, I'd be happy to take any questions that you have. Thank you.